Zenklusen, thank you very much for your valuable time. We are glad to have you here at IISER Pune.
My pleasure. I'm truly enjoying the time. It has been a very, very, very fruitful meeting.
So, cancer is the world's second major cause of death according to the latest World Health Organization study. And in 2018, it was responsible for an estimated 9.6 million fatalities. Cancer causes about 1 in 6 deaths. In this context, can you please tell us about the significance of the Cancer Genome Atlas project or TCGA?
Well, I think one of the reasons that cancer causes so many deaths is because we don't have therapies that are appropriate for the disease. Our understanding of the disease was very, very limited, and the reason it was limited was because we did not know what were the parts of the cell that were being activated or deactivated in order to produce a tumor. There were theories on the mechanisms, and we knew what functions were there regulated, but we really didn't understand what were really the gears that produced that, because we didn't have the list of genes. And so after the human genome was completed, now we have a parts list, if you want. And so the idea of the Cancer Genome Atlas was to use what we had learned in the genome and use genomic technologies to now see what are the players that are important in the different cancer types. And so that's why we characterize 11,500 plus cases in 32 different tumor types. And we did it in different tumor types because different tumors act very differently. And so the whole idea was try to see what makes the different cancers similar or different at the molecular level, because that also will allow you to figure out which players are good targets for therapy. Okay. And also it will allow you to classify the cancers correctly, because one of the things that we have been doing was classify the tumors depending on where in the body were located instead of how the tumors were behaving. Now, in fact, we know that, for example, there's one subtype of breast cancers that looks a lot more similar to ovarian cancers than the other breast cancers. And so therapy for that particular subtype must be different from the ones that are more common among each other. But until we did this project, we did not know this. And so that is what the advancements in the clinic are happening through TCGA, because now we have a better understanding of what makes the tumors tick, basically.
Okay. So, Dr. Zenklusen, is it true whether the incidences of cancer has increased over the years, and what causes cancer?
That is something that I get all the time. Okay. And I always say, no, it's not. What has happened is that we are better at detecting cancer, okay, and we are better at reporting it. And so now we have a more accurate picture of what is the incidence, okay, than that we had years ago. Also, one of the things that I frequently hear is, cancer is a modern disease. It didn't used to happen in olden times. And my answer to that is, yeah, because people in olden times died at 40 years old from an infection. Cancer is a disease of old age. And so when the population starts aging, you start seeing more incidents. But the overall adjusted by age incidence has not changed. Okay. We are seeing more because humans are surviving longer. Okay. And so, you got the opportunity to see the cancer appear that you didn't have in the past.
Okay. That's interesting to know. So we would like to know, how did the idea of this project came into existence? How did this project actually begin?
Before I went into TCGA, I used to be the senior scientist in oncogenomics in the neuro-oncology branch at NCI. And we had a project to characterize gliomas, glioblastoma multiformes, astrocytomas, oligodendrogliomas, 1,000 of them, using genomic level technology, but very primitive genomic level technology, not what we have today. And we actually did that project. It was called the Glioma Molecular Diagnostic Initiative. And we put together a database called Rembrandt, which is still being used even if the data is done with older technology, because the data is good and reliable. And so when that was published and created, NCI came to us and said, well, do you think we can do this with other tumor types? And the answer was, yeah, of course, the technology existed. And the interest got piqued because our project showed that you could get new targets for therapy if you do the study at the molecular level. And so NCI said, well, you have done that for brain tumors, let's do it for others. So that's the genesis, the origin of the project. One of the questions that I get frequently is why GBMs, why glioblastoma multiforme? Because although it's a absolutely lethal tumor, has a low incidence as tumors go. And my answer always is, oh, we had the samples because we had done the project and we had them in the freezers. And so that's why GBM was the first one. But also that particular work in GBM demonstrated that, there's something to learn by doing genomics in cancer.
Okay. The TCGA project has collected data from more than 11,000 cases across 33 different tumor types. What have the steps been?
Well, the steps are always the same. Some of them were more complicated at some time, and as the problems were solved, the new problems arose. But basically, the first thing you have to do is find your samples. And so we did contact hospitals that were known to be specialized in particular tumor type when we were there, and ask for the samples, because the samples is not just the tissue, it's the tissue, all the clinical data for the patient, pathology reports, histology slides. And so that at the beginning was the worst part of it, getting the samples of good enough quality to match the platforms that we're using at the time was the really difficult part. Then as the projects advanced and the publications started appearing, people really wanted to start participating in the project. And so getting the samples became less complicated. But now our problem was we had a lot of samples and we didn't have a very robust pipeline to do the characterization. So we had to expand the pipeline. And the pipeline basically is the samples go to a center that processes those samples and extracts DNA and RNA. And then that DNA and RNA is sent to sequencing centers that produce the sequence. And then that sequence gets deposited in a repository where the analyst can pull the data, do the analysis, and finally we produce the publication. So as we were solving the problems of different parts of the pipeline, the next step in the pipeline became the bottleneck. So first it was the sample, then it was characterization, then we didn't know where to put the files because we didn't have a repository big enough. When we solved that, then we needed to have the transportation of the data to the analyst because the internet was not big enough. These files are so big that take a very long time to do. In fact, at a point we were transporting data by shipping actual hard drives with the data, and then having enough expertise and good algorithms to do the analysis became the other bottleneck. At this point, we have none of those. And so all the new projects that we are doing are going swimmingly well because we basically have now a robust pipeline that produces all of this.
Could you please tell us how the quality of the data is ensured?
Well, we have a lot of quality checks along the line. So, for example, when we extract DNA and RNA, we run certain tests to make sure that the quality is good enough. The vast majority of the time it is, but when things are of not the correct quality, we don't lower the bars. Because sometimes what happens is you have so many samples and you need a little more, and then you go, well, maybe these ones will be okay. We never did that. Because if you start with something that is not of the quality of the rest of the data, you're going to have a separation of the data when you do the analysis. And so there was no point, there was a waste of effort. And so some of the projects have less samples than it would have been ideal, but all the material was all of the same quality. And then when we produce the the sequencing data, there's a lot of quality checks that are built into the systems. And again, sometimes you run the samples and you look at the data files and you realize that, oh, this is not good enough, or there's not enough coverage, which can be solved because you can run the machines a little longer and then now you have coverage. But if we see that the library was not constructed correctly, well, we go back and we redo the library. And so that's why people like the TCGA data, because it has been really nicely curated. And so you don't have to worry to see if there are batches or there are errors in the data. You know that the data is reliable.
Okay. So like the goal of this project is to diagnose and treat cancer patients. Am I correct? So how this scientifically generated data set will be applied in order to diagnose and treat cancer?
It's diagnosing and finding targets for treatment.
Okay.
We don't take it all the way to the treatment side. We suggest targets that could be used for treatment, and then the tools need to be developed in order to have the treatment. We don't create drugs. We don't run clinical trials. Okay. All that is a step that comes after our work.
Okay, that answers my question. So like if to a 15-year-old child you have to explain what molecular changes occur in cancer cells, how would you?
I have to do that on a frequent basis. So what happens in the way cancer happens is, let's go one step before. So we all get tumors. Tumors is an accumulation of cells in a cellular compartment or an organ that are more than what the organ of that cellular compartment needs. Okay. And so, for example, a wart in your skin, that will be a tumor. You have more cells there than the skin needs. That evagination of the skin shouldn't be there. But although that's a tumor, that's not cancer, because it's not malignant and it's not going to kill you in any shape or form. That wart could become malignant if it acquires other functions that are necessary for the tumor to become malignant. And these functions are: the cells need to continue replicate. The cells need to not listen to the signals from the rest of the body that tell them, don't replicate anymore, we have enough of you. Okay. The cells need to evade cell death, which is a programmed system to maintain the homeostasis, maintain the number of cells that you have in the compartment. The cells need to learn to invade the surrounding tissue, and they need to learn to metastasize. And so when the cells learn to do those things, then you have a tumor, and you have a very malignant tumor that ends up killing the patient. Normally, the primary tumors, the primary tumors don't kill the patient, except for probably brain tumors. But it's the metastasis, the one that does the deed. And so it is a fairly long process. And that's why also we always say that the best way to treat cancer is to find it early. Okay. Because if you find it early at a stage in which has not invaded the surrounding tissue, then through surgery, for most types, you can remove that tumor and the patient can be cured.
Okay. Is there any methodology from which we can detect which benign tumor can get converted into malignant in future?
Not yet, but that's our next projects. Okay. So our next projects are looking at samples that we're collecting during clinical trials. And there are certain tumor types, for example, lung or colon, that if they are found early, stage 1 and stage 2, they get removed surgically. They get treated with chemotherapy and maybe some radiation. And out of all the patients that are in that group, about 40% of them are cured. Okay. Totally cured. They're done. They don't need to be seen again. And about 60% of them will have a recurrence, and that recurrence will end up killing them. Okay. And right now we cannot differentiate between the two groups. Under the microscope they look the same. And so well, some of the projects that we're running are to try to figure out what is the biology difference between the good group and the bad group. Reasons are many. If we can determine that you are in the good group, once we finish with your therapy, we can say, you know what, let's see you every year for the next 3 years. And then after that, you know, you can come and have dinner with me, but I don't need to see you as a patient. While the ones that are recurring, and now we know that they will be recurring, we can follow them up a lot closer and try to change an acute disease into a chronic disease, basically. We may not be able to cure you, but we may be able to manage your disease.
Okay. That's interesting to know. So, how was it like to coordinate the effort of large teams as TCGA's director, and what were the difficulties?
Interesting is a way of saying. It is very interesting because every single so the group of people that we have for the molecular work, it's always the same group because it's a group of people that we have paid under grants or under contracts. So these are our people. The people that changes from project to project are the clinical side because they are the experts in that particular tissue type. And so I think it's very interesting because you get to meet a lot of new people. You get to work with very different personalities. Sometimes it's an enjoyable task, sometimes not so much. But it's also, I like the challenge. You know, if the work is always the same, I get bored. And I can say in TCGA, not 2 days are the same. And also is something that for me is very rewarding because I know that what we are doing is going to make a difference in the life of the patients. And so that's the reason why I started working in cancer is because I do care for the patients.
So, what are the ethical concerns related to this project?
Well, there are several. I think the more important one is maintaining the privacy of the patients because the clinical data is very easy to anonymize. You know, we can make disappear all the marks that say this is the patient that had this tumor. It's fairly easy to do. People think that it's a complicated event. It's actually fairly easy to do. The problem is when you are sequencing the entire DNA of a person, you have the person. And so if anybody had a stretch of DNA of that person and had the sequence, it would be able to identify it. And so what we are doing is we are not making the raw data available openly. You can access it, but you need to ask permission for it. And you need to be granted permission. And while getting the permission granted, you promise that you're not going to give the data away, because otherwise we lost control. But also that you're not going to try to identify the patient. And so that way we can help cover the patient. One of the other problems is that sometimes when we are doing this analysis, we discover things that are not of the tumor, but of the patient. And as such, and these things that their family members may have an impact on their health. And so now we have the obligation to let the patient know, but everything that we do for the characterization is for research only, it's not for treatment. And so it's difficult because we have to go through the treating physician. The treating physician has to order the test again. And so that is a complicated thing. And at the beginning, we were not returning results. But now we have been returning results when they are relevant to the treatment of the patient and their family. Okay. So that probably is the major problem. And sometimes people just don't want to know. You know, and the patient has a right to say, no, I don't want to know. And so that is quite a complicated issue.
So at a personal level, what was your motivation to do cancer research?
So My wife and me, we do both cancer research. We are both chemists. She has not had a brush with cancer in her family. And so from time to time when we were doing our PhD, she will say, you get frustrated and tired and all of that. And so she will go, I don't know why I'm doing this. And I immediately said, well, I'll show you why. I will grab her, go to the hospital, sit in the hospital and say, look at the people going by. Yeah. And she didn't know this, but one day she said, it's like, why I am the only one that needs to come from time to time to see the people going by. You never question yourself. Yeah. And the reason is because I had a cousin of my mother who was about 5 years older than me. So he was a lot younger cousin of my mother. Got married, came to India in her honeymoon, enjoyed it terribly, but she started having a very persistent cough. And when she went back to Argentina, the cough kept coming. She goes to a pneumonologist. They could do an X-ray plate, and there she had a sarcoma growing around her lungs that was pressing on her lungs. And so she started treatment, but the treatment was going nowhere. And she lived in a provincial city, which had less treatment options than you find in the capital, in Buenos Aires. Yeah. And so we were living in Buenos Aires, and we were the family that she knew. And so she basically came to live with us while she was getting treatment. And she had 2 years treatment, and she went down until the moment she died. She went from this very pretty young lady that she was a chemist too, that was supplementing her income by being a model for Elle and Vogue. So, she was a really, really pretty young woman, to be this skin and bones person that I used to take her to the treatments. And at the beginning, I helped her up the stairs to the hospital. And at the end, I would just pick her up like nothing, like a little kid, and to get treatment. And then finally, when she died, I said, well, this is a horrible thing. And now it's personal. And so I still have her picture in my desk. And whenever I get frustrated with anything, I look at the picture, and the frustration goes away, because my frustrations may be minuscule compared to what the cancer patients go through. And so that's something that stayed with me and still stays, and it will stay until the day I die.
Okay. Would you share with us your journey as a researcher from a doctoral student to becoming a director of TCGA?
Well, people that look at my CV thinks that everything was done in purpose, because it looks that way. Okay. Totally by chance. So I will go even before a doctoral student. So nobody in my family ever did anything in science. They all have businesses and work in with tourism, mostly in Switzerland. And I, not having had experience with science, I always liked architecture and design, so I was thinking I was going to be an architect. Until I got my first course of chemistry in high school. And I had a genius of a professor, Dr. Bandinelli, who was very clear and very engaging. And so after the first class of chemistry, I said, well, this is actually very cool. And so I decided to be a chemist. I became a chemist, and I was working in protein chemistry. And then this cousin Susanna died, and I said, well, I want to do cancer research. And so when my wife and me got married, we moved to Texas to the MD Anderson Cancer Center to get a PhD at the MD Anderson. And being that we were outside the States, we applied to all the regular universities that you think, Harvard, Yale, Stanford. We got accepted everywhere because we had a really, really good GRE, But we decided to go to MD Anderson because we wanted to do cancer. And so you go to the place where they have the resources. And so both of us, we were in the same laboratory. And our advisor, a very wise man, Dr. Conti, said, well, you know, you guys are married. I don't want you to have projects that are very close to each other because I don't want too much collaboration or competition. He said, I don't want to be the reason why you get divorced. And so my wife took the main line of the laboratory, which was cell cycle and cancer biology, and I took a line that was nobody wanted to do, which happened to be cancer genetics, finding tumor suppressor genes in chromosome 7. And so after I finished my dissertation, I had a map of a part of chromosome 7, and at that point, the Human Genome Project was launched. And we needed to do the maps of the different chromosomes, and Eric Green was a new principal investigator at the Center for Human Genome at NIH, which was just founded 2 years earlier. And I went to postdoc with Eric because he was doing the map of the chromosome that I needed, and so we helped each other. And the funny thing was, the first section of the genome ever to be sequenced was the section that I was working on. And the reason was because I already had a map, and so I had to populate them with the bags. It took me 4 months to do that. We sent the bags to sequence, and they were sequenced. And after that, I decided that genomic technology is fun, but I wanted to go back to cancer. And so an opportunity arose to be at the neuro-oncology branch, and I did that project that I told you about. And that project basically launched TCGA. And then when I got to the point that I didn't want to be attached to the lab anymore, the opportunity arose to be the director of TCGA. But when people look at my CV, it looks like, oh, all the steps are the correct steps. Yeah, they are, but they were just by chance. Basically, you know, the opportunity came in front and I grabbed it. So, you know, to any student that is thinking about career, what I will say is, you know, get the most basic possibly training, because that is very flexible. And then as the opportunities go in front of you, because they will, don't wait too much and grab them, because that's the way you progress, basically.
So we would like to ask a final question. What we all want to know is how long until we can cure cancer?
This answer is not going to be pleasing. Never. Because one of the things that I get asked this all the time. When are we going to get a cure for cancer? And my answer is never, because cancer is not one disease, it's 300 diseases. So asking for a cure for cancer is like asking a cure for hepatitis, the whooping cough, and dandruff. Okay, they're things that are totally different to each other. Each one of them has a different treatment. Some tumor types are less pernicious and less variable than others. Those we are going to cure. But there will be some tumor types that are really incurable, because you attack whatever is making them aggressive at the time, and whatever cells survive will learn to use other mechanisms to be aggressive again. And so the hope for that is that we can transform them, as I said, into chronic diseases. We are going to treat whatever you have at the moment, and when cancer comes again, we are going to identify exactly what that is, treat that, and then eventually you're going to have a relapse again, and we're going to identify what's the target and treat that. And so it will be a series of treatments, which means that we will be able to keep you alive and well for 40, 45 years, and you may die of a cold, or you know, a bus runs you over, or something like that, but you're not going to die from cancer. Basically, some tumor types, some cancer types, will transform into a disease similar to, say, diabetes. You know, we don't cure diabetes, but if you treat it and you maintain it, you can live a good and plentiful life. And so that's the goal for those type of tumors.
Okay. Thanks a lot for your time.
My pleasure.